Welcome back, folks. This is the quick and dirty version of cell membranes. Cell membranes are broken down into two basic categories. Let's put them down on the slide. We'll be talking about the lipids and their chemical and physical properties and how they impact the membrane. And then we're also looking at enhancing that functionality by incorporating proteins into these membranes too. Where do we begin? Let's talk about a cell in general. A cell will have a membrane called the plasma membrane that encloses it. All living things have a plasma membrane. One fascinating thing is all membranes are more or less based on the same physical architecture, although their chemistries might be somewhat different. When it comes to eukaryotic cells, we have a lot more membrane to contend with. We find that there's membrane inside the cell in various places. So the nucleus has a double membrane, believe it or not. Other structures inside a plant cell that would have double membranes will be your mitochondria and your chloroplasts. In fact, the chloroplasts have a third membrane deep inside. Other structures will be your Golgi, your endoplasmic reticulum, your vesicles, your lysosomes, your peroxisomes, your transport vesicles, right? So there's a lot of areas in which there's membrane. In fact, there's more membrane inside a eukaryotic cell than there is on the outer surface, the plasma membrane. Certain properties of membranes are very important. Membranes have to be flexible. If the membrane is not flexible, then it's unable to conform to the requirements of its job. So that's what we find. Animal cells are more flexible than plant cells or fungal cells. So that has to be then contended with in the physical and chemical properties of membranes. Regardless, let's just stick to what's common to all membranes. All membranes are built upon a foundation. And the foundation is that you have two layers to the membrane. And you have water on either side. And that will influence the composition and the construction of the membrane. What we'll learn in a few seconds is that the building blocks of these membranes, the main building blocks, are limited to two components, right? They're going to be lipids. Lipids are substances that do not dissolve in water. And that's important because the interior of the membrane will be hydrophobic, water repelling. And the second major component are the proteins, which give the membrane additional functionality. So keep those two things in mind, and you shouldn't go astray with this particular chapter, right? And of course, sugars and other molecules can be sprinkled onto and around that membrane to give it additional functionality. The most important thing is that when we look at any membrane, whether it's the double membrane of the nucleus or the single membrane around the endoplasmic reticulum, each membrane is divided into two halves. And one half is called a leaflet, right? So that's the top leaflet, and then we have the bottom leaflet. And that's important because as we study the lipid molecules next, you will see that they sit with their tails facing each other and their heads facing the world around them. So that double leaflet is the feature that gives the membrane its water repelling functionality. And that very simple region, which is about 25 to 50 atoms thick, that's all it is able to keep the inside chemistry of a living cell different to the chemistry outside. It's actually fascinating, right? So once you study this in a bit more detail, you will come across various models that propose how these membranes are constructed. And the favorite, and the one that's gathering the most data, is the fluid mosaic model, which says that the components of the membrane, these lipids, these proteins, are able to move around within their leaflet unhindered in most cases. So this particular lipid molecule can move in either direction. or even towards us and away from us. So it's free to move. So that's the fluid part. And the membrane itself is very flexible, like the surface of the ocean. It can be contorted and flexed in various manners. So that's very important to know. So let's concentrate on the building block, the primary building block, the lipid molecules. So where do they come from? Well, in your diet, you have food, and some of that food consists of fats. So those fats are basically lipids, right? So lipids is the better name to use than fats, because fats indicate one type of lipid, where lipids is a general term. So the lipids enter your body, and they're delivered to the cells via your blood. And in the blood, they're transported as simple structures that resemble a jellyfish. So here we are. So the head is made of a water-accepting substance, and the tails are made of fatty acids. The tails don't like water at all because they're made of carbons and hydrogens. And as you know from the chemistry chapters, carbon and hydrogen bonds, the covalent bonds that hold them together, they're non-polar. So the whole tentacle of this jellyfish, the triglyceride, is going to be water-hating. So the whole structure is known as a triglyceride, right? Triglyceride. In some books, they call them triacylglycerides, but they're talking about the same thing. So the head is included in that definition, as well as the three tails. So what is the head made of? The head is made of glycerol. Glycerol is a 3-carbon molecule that has oxygens embedded in it. So that makes the top of the molecule water-liking, the bottom of the molecule water-hating. And that's what phospholipids, it's not wrong word, that's what the building blocks of phospholipids are. So once this substance is delivered inside the cell then manipulates that by removing one of the tentacles and then replacing that with something else. And that something else normally contains a phosphate group, right? And then something else is added on the top of it. So you can see quite clearly we're going from a sea creature to a land creature, which looks like an emu. So these are called phospholipids. And it's the phospholipids that are the wonder molecule from which all membranes are constructed. So phospholipids is your main foundation, the brick for building your entire cell membrane house, okay? So let's go one step further. Let's look at the nature of these phospholipids. So if a molecule has a region that likes water and a region that hates water, then that molecule in chemistry is known, a single molecule that likes water one end and hates water the other end. That molecule has a name, and this is the main name that you should be using whenever you talk about membrane properties, right? So amphiphatic, right? So amphiphatic. So a molecule that likes water one end and doesn't like water the other end is amphiphatic. So these phospholipids are amphiphatic, and when you put them together in an environment of water, they will automatically take up a structure that has the least energy. So if you have a bucket of water and it's full to the surface and you introduce into it these fat molecules, they will generally float at the top because lipids are lighter than water. They have a lower density. So the density of lipids is less than water, right? So they'll float at the top. And if we magnify that view at the top, you'll find that their tails are sticking up and their heads are pointing down, right? Like that. And the water will be down here. Again, that goes back to the previous slide where this part of the molecule here likes water and this part does not like water. So this is the most stable, energetically stable configuration that they can adopt. Now, if you were to push these lipids into the water and seal the surface so that there was no air in the container, then these lipids will take up a different alternative structure that's more stable. And what they will do, they will form spheres. And the spheres will have the heads pointing up and the tails pointing towards each other in the center of the sphere, like that, right? So there's no water inside the sphere. It's just made of carbon hydrogen tails, the fatty acid tails. So these structures have names, and we need to know what they are called later on. But for now, let's go to larger spheres. So if we agitate the water and then get these molecules, which are not connected to each other, to rearrange, they can form a larger structure which has water on the inside and water on the outside. And in this case, they will rearrange themselves so that their heads are facing the water on both sides and their tails are facing the center for both layers. So these two layers are called the two leaflets. So if you go back here, we call them the leaflets, right? So now you're beginning to understand how a large structure with water inside and water outside will form a shell. And that shell is made of a bilayer of lipids, the upper leaflet and the lower leaflet. So that's very important. And if you look very carefully, if you have water inside and you have water outside, and then you have a shell of lipids, a bilayer, then that's resembling a cell. So that could be a prototypical cell that formed on the planet as soon as lipids became available. At that time, the oceans were full of water. And so it was a watery world, right? So these are the building blocks of lipid membranes. Now, when you look at a structure of a phospholipid, you can draw it just with a circle to represent the head. But the heads can be of different types. And in fact, the tails can be of different types too, right? So these tails are called fatty acids. And let's talk about them first. So some tails can be longer than others. So when these are made somewhere inside the cell, they can be constructed of long tails versus short tails, right? And the length of the tail will impact the functionality of the membrane, the physical and chemical properties. But that's one thing, right? The second thing is that the tails can be straight or they can be bent or kinky, as some books call them, right? And the number of kinks that they have will impact how closely their neighbors come when they're stacked together. So if this is one phospholipid and there's another one with a kinky set of tails, right? Then the next neighbor will be pushed further out. And that's going to reduce the density of this particular fat compared to a fat that's made of straight-tailed phospholipids, which can line up really, really closely and increase the density. So fats come in different flavors. That means that they come in different chemical compositions. One of the properties that the fats have is that they can be stacked together to make solids at room temperature, or they can be stacked together to make oils at room temperature. So this is what you find in animal fat. This is what you find as a fat in plants. Okay, so that's a good analogy. But let's concentrate now on the heads. So let's draw that structure again. And we said that the tails can be long or kinky. So that's two properties that you need to remember. Let's now concentrate on the head. So the head can be made of various building blocks. So the most common is called phosphatidylcholine, phosphatidylserine. What does that mean? That means we have, let's amplify that a bit further. So at the bottom here, the bottom part of the bird, we have the glycerol molecule, right? So it's normally glycerol, it can be exchanged with something else. So we have the 3 carbons, and the bottom 2, I should say, not the third, the bottom 2 are attached to the fatty acids, right? So those will be the fatty acids, okay? And the top part of the head is then able to bond with other structures. So that third carbon there normally turns around its bond, and it can now bond with various things. And the most common is a phosphate group like this, okay? And the phosphate group will then give that part of the molecule a very negative charge because of the high electron pulling potential of the oxygen. And then the top oxygen can be attached to something else to make the mane or the hair of the bird, right? If you put it that way. And that can be various things. It could be amino acids like serine. It could be more nitrogen-containing compounds like choline. Or it could be other types of molecules like sugars, right? But what you're looking at is the general architecture of what a phospholipid looks like. So that takes us to the final part, that all membranes are not made of the same phospholipid, right? So you could have a 50% proportion of this type, and you could have a 10% proportion of that type. You could have a 5% proportion of that type, et cetera, right? So that will now give that particular leaflet a particular chemical identity that will then influence its behavior. And the other leaflet, of course, could have any combination of these, because each leaflet is made according to a certain recipe that the cell knows about. So the two leaflets of the plasma membrane, or any membrane inside the cell, would consist of these phospholipids arranged in that particular fashion. Okay, there's a nice video on the website that talks about where these lipids are made and how they're delivered to their destination. So we don't have time to go into that in this particular video lecture. So let's move on and let's talk about proteins now, right? Let's turn our attention to proteins. Oh, I forgot to tell you the name of that structure there, right? This structure here, where you have water outside, let's draw it again. You have water outside and water inside. That's called a liposome, right? Liposome. So liposomes are very, very important. This is called a micelle. This is called a liposome. So remember the difference, right? A micelle has no water inside the core. It just got hydrophobic tails. And a larger structure called the liposome has water inside, and therefore it forms a bilayer of lipids. One thing that we need to finish off with the lipids that we must get out of the way is what can pass through the membrane. So let's draw the membrane. Here's the membrane. And again, it's a double leaflet. So let's put a couple of these phospholipids in place so we know what we're talking about. So the question is, what kind of chemistry can pass through that membrane? Well, the first thing is small. Small chemistry, right? Molecules, individual tiny molecules, or maybe even individual atoms like argon or neon. So if you're small, you have the potential to pass through, okay? So examples of that will be oxygen and carbon dioxide, right? So these are very small molecules. Number two, you have to have a non-charge, no charge, okay? That means you can't be polar and you can't be an ion. So hydrogen ions, even though they're the smallest chemistry, even smaller than oxygen and carbon dioxide, they cannot pass through. They cannot pass through the membrane because of that positive charge, right? So any chemistry that has a charge, positive or negative, will be prohibited from passing through the lipid bilayer of that membrane. Large molecules can't pass through. So glucose will be too large. It contains too many atoms. It's too big. It can't squeeze between those phospholipids, okay? So generally, small, non-charged molecules can pass through the membrane. Now, water is a weird one. Water can pass through the membrane, even though it's a polar molecule, because of its size and its concentration, right? So it sort of fights its way through to a reasonable degree. So water is an odd one. But what cannot go through are large molecules and charged molecules. So there's one last thing. So molecules that can dissolve in water, lipids, other lipids like hormones, lipid types of hormones. So they, because they can dissolve in there, they can pass through. So if you're a hormone of the lipid type like testosterone, you would have no problem approaching the membrane and then just making your way through, right? Because the interior of the membrane is friendly to you. You don't like water, it doesn't like water, you can pass through. So remember these properties, they're very important. But now you see a conundrum. I told you earlier that glucose cannot pass through. Amino acids cannot pass through. So the major food molecules cannot pass through. So how do you feed the cell? And the answer is, you get the assistance of proteins. So when proteins come along, what they can do, they can change the nature of the membrane to assist you. And proteins are fantastic. So we draw the membrane like this, and then we can have various classes of proteins that can sit in various places, helping the membrane perform additional functions, right? Other proteins, let's choose a different color so we don't confuse the two later on. Other proteins are just attached to the outside and the inside, or just one or the other, right? So there's a difference between the blue and the red. So the blue ones are considered to be integral. They're held in place by more than light forces, right? Whereas the red ones, they're only held in place by non-covalent forces. So the red ones can be easily removed with a gentle treatment. So they're called peripheral membrane proteins. So if you can easily remove them, they're peripheral. If they're hard to remove, then they're going to be integral. So remember that. So those are the two major integrations with the membrane, right? But let's go to functionality. So functionality, proteins enhance the membrane's functionality tremendously, right? Some of them can be for transport. That means they can allow chemistry to pass from one side to the other. So this is what glucose would use. Glucose would use its own customized protein called GLUT, and that particular protein will only allow glucose to enter and leave the cell depending on its concentration, right? So if the concentration of glucose is high in the blood and low inside the cell, the GLUT turnstile will allow glucose and only glucose to enter the cell. If the concentration of glucose is high inside the cell and low inside the blood, as happens in some cases with liver cells, then the liver cells will release their glucose into the blood using the same turnstile that runs in the opposite direction, okay? So this is how you import amino acids and large food molecules, and also get rid of waste products too, okay? So transport proteins are very, very important, and they're littered in many varieties in every membrane that's natural, right? So that's one thing. Then the next thing is anchor proteins. So just like you have anchors in a ship, anchor proteins hold this cell in place. So the classic example is on your teeth. You have lots of bacteria that cause tooth decay, and they release well, they have a protein in their surface that they use for attaching to your teeth, right? So those would anchor the bacteria in place so that they don't get washed out by your saliva. And again, inside your body, you have skin cells that are anchored to each other. So when I pinch my skin, they don't come apart because the force is dissipated in all directions between these cells, okay? So anchor proteins hold things in place. The next one are your receptors. Now, receptors would also be normally going right through the membrane. What receptors do, they bind some chemistry on the outside and they change their shape on the inside. And then that internal change is recognized by something that's watching the inside of that protein, and that causes the cell to change its behavior. So receptors can detect the world around them, right? Very important. And then we go on to enzymes. So if you have substance A floating around inside the cell near the surface of the membrane, then it can be a substrate for this enzyme, and it will bind to the active site, and then it will be converted to a product. So enzymes can be attached as part of the membrane rather than floating around freely inside the cytosol, okay? So the membrane can serve that function too. In fact, the functions continue again and again other than this, right? So I told you that hydrogen ions cannot get into the cell by themselves. Well, there'll be a special hydrogen ion transporter that will pick up hydrogen ions, whichever side they're highest at, and then transport them to the side at which their concentration is lower. And that will be called facilitated diffusion. Why? Because you're facilitating it with the help of these proteins rather than just simple diffusion, as you saw earlier with the lipids, okay? So proteins really do enhance the functionality of these membranes tremendously. So you need to remember that, right? Now, very quickly, just to get a bit more detail-y, how do these proteins coexist with a membrane where it has heads that like water, the circles on the phospholipids, and the tails that hate water? Doesn't this contradict that? Not entirely, because nature is beautiful. So the genes that are responsible for making all these membrane proteins, they have evolved to have the right amino acids inside the protein interacting with the right part of the membrane. And this is a fascinating stuff, right? So let's change the color again, and let's use a green color. No, green doesn't show up that well. Let's use orange. So suppose we have a protein, a transmembrane protein, and the word transmembrane means just like transatlantic, right? That you go from one side across the Atlantic to the other. So transmembrane means a protein starts on one side of the membrane, goes right through to the other side, right? So here we have a transmembrane protein. And this protein has to coexist inside that membrane. So we can see that certain parts of the protein should be here, especially from this point down to about here, where the heads of the lipids are, right? This part of the protein all the way down here should be hydrophobic, right? So its amino acids should like hydrophobicity, right? They don't like water, right? So those amino acids are used to make a spiral that spirals through here to the other side. Not to the other side, to the next hydrophilic. So from this point onwards, let's change the color once more. Let's use green now. So from this point onwards, the amino acids will all be mostly water liking, right? Hydrophilic. And same thing up here. Once you get to the head region, then the amino acids that are making up the polypeptide, the protein, will become water liking as well. So you can see this particular protein has three zones: a water liking, a water hating, and a water liking. Just to finish off the story, the most common type of protein that exists in the membrane passes through the membrane 7 times. That's the most common. So let's put that in place very quickly, right? Let's use the same orange color and let's draw it over here, right? So the protein may start inside the cell, goes through the membrane, comes out, makes a loop, comes back, makes another loop, and it does that 7 times, right? I think I got it 7, 2, 4, 6, and then 7, right? So that single polypeptide, that single protein, has to have all these properties. So this part of it, that chain is going to be water hating. So is this part, so is this part, so is that part when it comes back in and out. Oh my God. So you see that? And then the loops will be water liking. So this loop will like water, this loop, that tail, that loop, that loop, that loop, and that tail. And this protein here was made by a single gene that knew which parts would be inside the membrane and which parts will be on the outside. The information stored in DNA is amazing, right? So the cell can do that because it has evolved those proteins to do so. So sometimes the zigzags that I put there, these ones in blue, they can form cylinders that spiral through the membrane, right? They're called alpha helices, right? So when you do the reading, you'll see that. And then of course, they don't make a 2-dimensional structure. They don't make a sheet like I've drawn here. They make a cylinder where these things are arranged in a donut-like shape, right? Like a circle. And then down the center will be a cavity, and that cavity will be free of phospholipid tails, and that cavity will then be available to pass material, specific material, right? So if this is designed for glucose, then only glucose will be transported. If this is designed as a receptor to detect light, the only thing that you'll find in the cavity will be vitamin A in your eyes, and you can then pick up light energy, right? Most of these have lids on their passageway, so that they can control what goes in and out of the cell, which is amazing. So proteins can come in very simple designs to very complicated, right? Just to finish the story off, if you are a protein that is embedded in the membrane itself, say, let's put that in red, say you're a protein that sits like this, and all you do is sit up here and then you go back out, right? So that's still an integral membrane protein because it's hard to pull that protein out of the membrane, right? Because of all its interactions with the surrounding chemistry. But in this case, because it doesn't go into the interior of the membrane, most parts of that protein are going to be water liking, but some parts are going to be water hating or water disliking, right? And so let's put those parts in. So we use blue for that. So the bottom of each loop that's facing now the tails would be hydrophobic, right? And the rest of the red would be hydrophilic. So these are very important things. And just to finish off the story, sometimes instead of using these alpha helices, these spirals of polypeptide, nature can use beta sheets. So if you go back to the chapter on protein structure, you will know that you can zigzag the polypeptide chain to make a sheet. And you can take a sheet, like a metal sheet, and mold it around into a giant cylinder. And that giant cylinder will then sit inside the membrane. Let's draw the membrane once more down here. So here's your membrane, and the giant cylinder will sit like that, going through the membrane. And that will be made of zigzagging polypeptides, right? Again, you have to have the right amino acids facing the tails, and then you have to have the right amino acids facing the water in exterior and interior. But this can form a giant barrel through which things can pass when the lid is open. You can close the lid to regulate that. So water, when it's needed in huge quantities, like I told you earlier, water has a sort of semi-permeable ability to pass through the lipid bilayer. But in this case, say in the kidneys, you need to regulate water in huge quantities. Therefore, you'll have this type of specialized protein called an aquaporin, right? And that's what aquaporins can do. Okay, so I think we're there. This is it. So this was the long and the short and the dirty, right? What else do you need to know? Well, let me think. Just one or two things, right? Very quickly, just to finish off. I said I was going to end there, but I'm not. I'm going to give you just two more things. Let's use this felt tip pen. Let's relay some information to do with strengthening the membrane. I told you the membrane is very flexible because of the fluid mosaic model. Because the membrane is very thin, it's not more than 50 atoms thick, right? Twice the width of or 2.5 to 3 times the width of a DNA double helix. So how do you stop the membrane from ripping, from falling apart? Well, cells are clever, right? So on the inner surface, they have proteins. And those proteins form a cortex, a scaffolding. And the proteins above, some of these, like this one, those are attached to the scaffold. And that anchors and gives shape to the plasma membrane. That's why red blood cells can be the shape that they are compared to the white blood cells, because their cortex is made of a different architecture than that of the white blood cells. So hopefully the long and dirty worked for you and that you got something out of this. Thank you so much. Bye-bye.